Cellular respiration begins with glycolysis, which occurs in the cytosol. Glycolysis is the process that converts glucose into pyruvate in a series of two phases, the energy investment phase and the energy payoff phase. In the energy investment phase, the cell uses 2 ATP to break apart the glucose molecule and 2 ADPs into phosphorus. In the energy payoff phase, substrate-level phosphorylation occurs, which adds phosphate groups from substrate molecules to the ADP produced in the energy investment phase, producing 4 total ATP. NAD plus is then reduced to NADH and hydrogen by the electrons released by the oxidation of glucose. This in total produces 2 pyruvate molecules and 2 hydrogen molecules. In summary, the breakdown of glucose produces 2 pyruvate and 2 H2O molecules. The 4 ATP formed minus the 2 ATP used in the results in 2 ATP, which can then be released. The 2 NAD plus the 4 electrons plus the 4 hydrogen molecules equals 2 NADH, which are then used to power later cycles and 2 hydrogen molecules.
Welcome to our lesson today on cellular respiration. We will be covering the
Pyruvate oxidation follows glycolysis as the pyruvate produced enters the mitochondrion in the presence of oxygen. Pyruvate oxidation consists of a series of enzymatic reactions that remove the carbon dioxide and forms more NADH from NAD+, which produces acetyl coenzyme A, also called acetyl-CoA, necessary in the citric acid cycle. Pyruvate enters the mitochondria where the oxidation of glucose occurs. Acetyl-CoA is produced when carbon dioxide is removed and NADH forms from NAD+. Two carbons are added to oxaloacetate from acetyl-CoA. The 2 carbon groups oxidizes and forms carbon dioxide. 3 NAD plus reduces to NADH for each acetyl group that enters the mitochondria. An ATP molecule is formed by the phosphorylation of ADP. 2 electrons and 2 protons are transferred to FAD to become FADH2. This cycle creates citrate, which then decomposes back to oxaloacetate and regenerates the cycle.
The oxidative phosphorylation process works through the pathway of electron transport, in which there is a sequence of electron carriers in the electron transfer chain, which drop in free energy as electrons travel down the chain. The electrons alternate between reduced and oxidized states as they accept and donate electrons. Each component becomes reduced as it accepts electrons from its uphill neighbor, then returns it to its oxidized form as it passes electrons downhill. Complex 1. Electrons acquired from glucose by NAD+, transferred from NADH, go into FMN or Flav protein. The FMN then returns to its oxidized states while it travels when it transfers its electrons to FES, an iron protein, which then transfers its electrons to ubiquinone or Q, which is individually mobile. Complex 2. Another source of electrons is FADH2, a reduced product of the citric acid cycle. FADH2 adds its electrons within complex 2 at a lower level than NADH, though they both donate the same amount of electrons, 2, for oxygen reduction. Complex 3. This complex oxidizes ubiquinol, which then goes into cytochrome B and then into another FES, and also reduces 2 molecules of cytochrome C. The electron is transported via these reactions onto complex 4, accompanied by the release of protons. Complex 4. The received electron is received by a molecular oxygen to yield a water molecule. This conversation occurs in the presence of copper ions and drives the oxidation of the reduced cytochrome C. Protons are pumped out during the course of this reaction. Three, two, one.
Glycolysis and the citric acid cycle only produce 4 ATP out of the total 30 or 32 ATP produced in respiration. The remaining ATP comes from oxidative phosphorylation powered by either NADH or FADH2. In this process, electron transport and chemiosmosis occurs.